Hello, my name is Tudor Oprea, and now we're going to talk about the data science aspects of illuminating the dark genome. As you probably know, data science has been used in several components of the drug discovery enterprise. And as we envision the three pillars of drug discovery, there is a significant component of data science, both with respect to diseases, for example, electronic medical record processing and nosology, as well as an increasing component of machine learning and artificial intelligence for EMR processing. Then we have data science in targets, and the critical component of that is how do we select targets for drug discovery. And last but not least, drugs, and there are multiple methods to identify novel therapeutic modalities using in silico methods. Essentially, Illuminating the Druggable Genome project is developing methods applicable in each of these three areas. With respect to data collection and the enterprise from the IDG Knowledge Management Center, what we're essentially focusing on is large-scale data set integration, both from the genome and proteome areas, as well as multiple resources such as Avimayan's Harmonizome, String, which is the largest protein-protein interaction database, etc. We had to develop our own drug target ontology with Stefan Schürer at Miami, and we are absorbing chemocentric data such as Campbell and DrugCentral, and I will get back to DrugCentral later. And once all this information is standardized, it's deposited into Target Central, and then it's accessible via the PHAROS knowledge portal. These are some of the data sets that we have digested and integrated into PHAROS since the last update, which was version 5. So in version 6, for example, we added information from the Cancer Cell Line Encyclopedia, String, and more recently PHIPSTER, which is a viral human protein interactions atlas, given the impact of the current pandemic. Overall, this is a growing data set which places emphasis on machine learning ready components. Key to our data annotation and understanding drug targets is a knowledge-based classification of proteins, which has 4 categories. T-clean, which are proteins that are annotated with known drugs. So it's the mechanism of action. This is how drugs act. And that was subject to a paper in 2017. And then out of those drugs that interact with targets, we annotated T-chem. And we are essentially looking at bioactivities as defined by the cutoff for approved drugs, and then trying to annotate protein target classes by bioactivity. So for example, for kinases, we have a cutoff of 30 nanomolar, whereas for ion channels, the cutoff would be as low as 10 micromolar. But by default, we assume that the cutoff is 1 micromolar. The other 2 categories of targets that we annotated are T-dark, also called the ignorome, for which we do a fractional publication count, which was pioneered by Lars Jensen in Denmark. We also look at gene references into function, and we also count antibodies available via antibodypedia.com. And then proteins that are not T-dark, not T-chem, or not T-clean, they typically end up in T-bio. And often they end up in T-bio where they meet 2 out of 3 criteria to not be in T-dark, meaning a higher publication score or more gene drifts or more antibodies, but also, for example, when there is a gene ontology molecular function experimental evidence code. So with that in mind, this is just a temporal evaluation of how target development levels have evolved over time. And we started counting in 2013 in December. And since that time, when we roughly had more than 9,000 proteins in T-dark, we are now approaching about 6,000. So there is clearly an increased effort in annotating proteins, and currently nearly 70% of the human proteome is not what we would consider T-dark. There is a much less dramatic increase when it comes to annotating drug targets. And we had some differences in how we defined drug targets, but the lowest was below 600, and there was just an increase of about 10% from 2016 to 2020. So why do we care about this? Well, it turns out that we're all facing this central dogma. We initially thought that, oh, we're just going to understand genes and we're going to look at the impact of the environment, and that should give us phenotypes. Well, it turns out that the equation is much more complex. We need to take into account epigenomics and metabolomics, pathogenicity, multiplicity of protein interactions, variable gene expression data, and frequency of alleles, and SNPs, etc. So it's all rather complex, and it turns out that we need to try to understand all these multiple interactions that help us travel from genes and environment to map traits. And this is clearly a data science effort. Take, for example, diseases. To put in context what I'm about to tell you about rare diseases, it's important to understand the leaf node concept. So, for example, when somebody looks for congenital anomalies of the cardiovascular system, one would assume that a congenital heart disease is one such node. Well, it turns out that congenital heart disease is an umbrella term that includes long QT syndrome and tetralogy of Fallot, etc., etc. So in short, what we're looking at to understand leaf nodes is those nodes that cannot be reduced to child terms when it comes to disease ontology. So we're essentially going to identify those diseases that form unique entities. Using that type of approach, what we've then done is to count the number of rare diseases, and in particular the leaf nodes in the disease ontology, which also are annotated as rare diseases. And for resources, we looked at disease ontology, Orphanet, GARD, which is the NCAT system, the NCI Thesaurus, of course, OMIM, which is probably the first such database in existence, and then the Mondo system from the Monarch Initiative. And with that count, we ended up revising the number of rare diseases from 7,000 to more than 10,000. This type of exercise has also helped us understand that there is a serious gap in translation between science and therapeutics. So, for example, if one monitors the number of publications in PubMed that have been dedicated to rare diseases over time, that number increases dramatically every year, whereas the number of approved therapeutics is still within hundreds when we count drugs approved for rare diseases in Europe and the United States. However, we think that there is hope. So trying to visualize the target space of rare diseases, if you look at the inner ring, about 7% of these targets that are associated with rare diseases are part of TICLI. And that's not to say that these are drug targets for rare diseases that can be easily manipulated with existing therapeutics. It is, however, a good sign in the sense that these targets we know can be manipulated in the clinical sense. And therefore, it's important to keep in mind that perhaps in the rare disease, this is a loss of function, and what we want to accomplish is gain of function. Whereas quite often in the therapeutic arsenal, we tend to develop antagonists and inhibitors. Nonetheless, this could be low-hanging fruit that could be studied immediately. All this data and more is available through our website, PHAROS.NIH.GOV. And there are three main components that one can explore. The most developed one to date is targets, since PHAROS is a protein-centric interface, but we also allow the user to query for diseases as well as ligands, which means small molecules that approve drugs. And according to Aaron Pawlik, who was one of the leaders of the IDG program until recently, PHAROS is considered to be the flagship of the IDG program. I also wanted to draw your attention on a topic called DrugCentral. So in DrugCentral, what we essentially try to annotate is a lot of information with respect to active pharmaceutical ingredients, which is what scientists and medicinal chemists, etc., they call that drugs. Whereas patients and medical practitioners, what they call drugs, are actually formulations of these ingredients. And we try to reconcile this as accurately as possible, and we offer multiple layers of annotation for each individual active ingredient. More recently, we have examined the FDA Orange Book to try to address the question of how many drugs could be available for repositioning. And if one looks at the Orange Book, there are the so-called on-market products and the off-market products. And of course, the off-market products, they go straight into the off-market bin. Whereas the on-market products, there's two categories, on-patent and off-patent. And with that in mind, we actually paid a lot of attention to the status with respect to both patent and exclusivity. Drugs can be covered by patents, but also the FDA can warrant exclusivity for formulations that may not be covered by an active patent. So when all that is sorted, we end up with 996 off-patent medicines, 237 that are currently on patent, and 320 which are off-market. And in this paper that was recently accepted in JCIM, we propose a strategy for drug repositioning. Another recent addition to DrugCentral is a platform that we call REDIAL 2020, which is in collaboration with a group of Suman Sirimula at UTEP. And we're essentially taking data from the NCAT's COVID-19 portal and develop multiple machine learning models to try to evaluate bioactivities of compounds from molecular structure. I'm going to spend the last minutes of my presentations with some thoughts on target illumination. As I mentioned before, we developed a knowledge-based classification system for proteins, which goes T-dark, T-bio, T-chem, T-clean. These are intended to offer overall criteria for illumination, but they're clearly slanted from a pharmaceutical drug development perspective. So in that sense, a simple cutoff for illumination is what we would consider T-bio. But if T-bio, T-chem, and T-clean are illuminating, then so is 70% of the proteome. The problem is, of course, we don't really know whether the function of a protein, just because it is simply in T-chem or even T-clean for that matter, we don't know if it's completely understood. So what I'm trying to say is that there are degrees of illumination, and one would consider perhaps that a well-illuminated protein is one that is well studied. It has hundreds of publications, and to the minds of many, the book has been written of those targets. However, I'm not really sure that there are a lot of examples of proteins that are completely illuminated. In my opinion, we are likely to discover novel functions for even the most studied proteins, just as if you change disease states or the tissue environment, the pH, etc., perhaps new associations will be uncovered. So in short, illumination is a context-dependent tag, and it can refer, for example, to the structure of a protein, a successful clinical intervention, a biological function, etc. So clinically validated targets, such as T-clean, are definitely on top of the illumination tree. However, we may have to rethink what would be the best way to assess illumination. In other words, understanding targets only in terms of clinical interventions might be short-sighted. We really want to understand biology. So perhaps use case for research reagents or a use case for biomarkers, which of course would have to be validated, might offer different angles of illumination. With respect to IDG, of course, what we're interested in as a consortium is moving targets out of the understudied category and making it more brighter for those targets, not so much for targets that are already well illuminated. Therefore, the challenge that we have is how to enable people to accumulate enough data to write a full-fledged proposal. Of course, the challenge is if it's a truly dark protein, there's not much to write about. So we really have to think about illumination in practical terms. We may have to reanalyze the entire set of data and redo some of the statistics and perhaps try to figure out whether this target development level, meaning T-clean to T-dark, is enough to understand these proteins. We may have to map a collection of use cases and try to examine different ways to classify knowledge and perhaps revisit illumination criteria. So in that sense, we really need community feedback to define illumination by example. It would be my preference that we get a comprehensive list, preferably up to 1,000 proteins, where scientists from different areas of different branches of science, different fields, would give us their view of what they would generally regard as illuminated. And then the hope is that we can identify from a data science point what these proteins have in common. So understanding what proteins get NIH funding might be, or EU funding for that matter, might give us a different way to look at this. And given that these data can be accessed backwards over a period of time, we might include a temporal perspective for this process. We still need to define the metrics to understand illumination. How much is known about the target? We currently have TDL, but perhaps it's time to revise that. And what is considered a validated level of evidence with respect to a particular protein? Whether we consider in vitro data to be sufficient, or we have to move in vivo, whether we consider approved drugs, etc. So please let us know if you want to participate in this exercise. With that in mind, I have to thank my wonderful collaborators. The thoughts on illumination were discussed with Lars Jensen, Jeremy Yang, Stefan Schürer, Noel Southall, Trang Nguyen, Tim Shields, Steve Matthias, Andrew Leach, Anna Waller, and Avi Maya. And of course, I want to thank my co-workers, Christian Bologa, Jamie Holmes, Giovanni Bocci, Praveen Kumar at UNM, Sorin Avram and the team in Romania, Suman Sirimola and the team in El Paso, and then Keith Kelleher and Vishal Shiramshetty from NCATS. Thank you, and I'm interested to hear your questions.